Now, meiosis is a very organised physiological anatomical event, and it's in stages. Now, when the cell doesn't appear to be dividing, we call that interphase. The interphase, when it doesn't appear to be dividing. There are things going on, as we'll see in a minute, but you don't really see it from the outside of the cell. Then there's the prophase. Then the next stage is the metaphase. Then the next phase after that is the anaphase. And then the final stage is the telophase. So you need to memorise these: interphase, prophase, metaphase, anaphase, telophase. And then there'll be another interface after that, usually. But in meiosis, this happens twice. So in meiosis, there's prophase 1, metaphase 1, anaphase 1, telophase 1. Then there's a short interface. And then this happens all over again. There's another prophase. This time it's prophase 2. Then there's metaphase 2. Then there's anaphase 2. And then finally there's a telophase 2. So it is somewhat of a complicated process. So let's start off thinking about it now. Now, if we start off thinking about the interface, what's happening in the interface? Well, in the interface, if you think about it because we know this comes from we know meiosis derives from germ cells. It's the germ cells that are dividing. These germ cells in the ovaries or the testis. Now, in the interface, what we've actually got here is 46 sets of chromosomes. So here's a chromosome here. 46 chromosomes, 23 pairs of chromosomes. So there's 46 chromosomes like this in this germ cell. It contains 46 chromosomes. I've only drawn one here. And the first thing that happens is all of the DNA. So this is the chromosome here. The chromosome consists of structural proteins and the DNA, the deoxyribonucleic acid that carries the genetic information. All of this DNA first has to replicate. So in the interface, this replicates. And what the chromosome actually does, if we just draw it a bit bigger, is it makes another copy of that arm there. So it makes a copy of that. So we end up with 2 of these. These are the same. It's just duplicated it. So it's duplicating that arm to make 2 arms. And these are called chromatids. So we have 2 chromatids here, and it duplicates that one there, again to make another 2 chromatids. So that chromatid is the same as that one, it's just been duplicated. And that chromatid is the same as that one, it's just been duplicated. So there's DNA duplication into 2 identical chromatids. Now that, when it's on its own, we call that a chromosome. That's a single chromosome, of which of course there are 46. And when these are divided, we call them tetrads. Tetrads, tetra for 4. As in tetraplegic, when someone can't move all 4 limbs. So now we have 46 tetrads. So in the interphase, there's a lot going on. All this DNA has been replicated. The whole process of meiosis in men takes about a month. It's quite an ongoing process. And as we've said, in girls, this process starts off in females, this process starts off in early fetal life. It doesn't complete till adult life, but it starts in early fetal life. So the DNA is uncoiled. Actually, in this stage, the chromosomes aren't as recognisable as this. It's only when we move into the first phase of cell division, the prophase 1, that the chromosomes condense and become recognisable as these tetrads. And this happens in what is often called the S phase of interphase. S standing for synthesis. It's the phase when the DNA is synthesized and replicated, completely copying itself. That's the great thing about DNA, this great double helix molecule. The molecule can divide and produce, and each half of the molecule can produce another perfect copy. It should be a perfect copy of itself. So that's the first stage, that's the interphase, where we have duplicated the chromosomes and duplicated these arms to make chromatids. So now we have 46 of these tetrads. They're all going to be different sizes because the chromosomes are different sizes. But all the way from chromosome 1 down to chromosome 46, we now have 46 tetrads in the cell. Whereas before, we had 46 individual chromosomes. Now we have 46 tetrads. So that happens during interphase, and that sets us up nicely for the next stage, which is the prophase, which we'll look at next.